you have to know about these steps. Okay, so that is, this cycle is very important because it is converting your ammonia. Okay, it is converting your ammonia into a less toxic form that is called as your urea, and then that is being excreted by your kidneys. Okay, so major is your ammonia conversion. Excess nitrogen is converted to urea and it's excreted by your kidneys. So now where is the urea cycle taking place? In your liver. Where is the urea cycle taking place? It's taking place in the liver, and that's basically both in the mitochondria, and it is also taking place in your cytoplasm, right? So now the cytoplasm of the mitochondria and cytoplasm of the liver cells, that is your hepatocytes. So you have to divide them into five main reactions. Okay, and the first is your ammonia coming and entering your hepatocyte, that is usually in the form of glutamine or alanine. That is usually in the form of glutamine or alanine. And then if you see, it is going to, once it is inside the hepatocyte, the urea cycle is going to begin in your mitochondria, and that's going to give us citrulline. Okay, and that's going to give us citrulline. So your ammonia is entering your hepatocyte usually in the form of alanine or glutamate. Okay, once it is inside the hepatocyte, the urea cycle begins in the mitochondria, and that is going to yield citrulline. Okay, so how is it going to yield citrulline? It is through two reactions. Okay, you have to remember these two reactions, because these two reactions, everything else is going to occur in the cytoplasm. Okay, after these two reactions, the remaining of your urea cycle is going to occur in the cytoplasm. So one, it is by your carbamyl phosphatase synthase 1, another is by your ornithine transcarboxylase enzyme. So your OTC, this is called as your OTC. Your OTC will convert your carbamyl phosphate into citrulline. Otherwise, your carbamyl phosphate synthase 1 will convert your ammonia and carbon dioxide, into carbamyl phosphate, and your CPS1 activity is controlled by N-acetyl glutamate. It is controlled by N-acetyl glutamate, and that is a allosteric activator. So this will go bind to the non-active site. It will go bind to the non-active site. Okay, on the enzyme to increase the enzymatic activity. That is why it is an allosteric activator binding to a non-active site to increase the enzymatic activity. So this reaction will require how much ATP? It will require 2 ATPs. Okay, so now your mitochondrial membrane. Okay, so I told you this is happening in the mitochondria. So mitochondrial membrane is only going to transport your citrulline into the cytoplasm when the final three steps is going to happen. So till now, is everything clear? Till now, the reaction takes place only in the mitochondria. When your citrulline enters your cytoplasm, we have three important things. One is your arginosuccinate synthetase. Another is your arginosuccinate lyase, and then we have arginase. Okay, so here this is going to convert your citrulline into arginosuccinate. Here it's going to require both your aspartate and your ATP. Okay, and then your arginosuccinate lyase is going to cleave your arginosuccinate to give us arginine, and it will also give us fumarate. I think that's given a little bit down. It will also give us fumarate. Okay, so fumarate is a metabolite that can be used in the which cycle? PCA cycle. And then next is your arginase, that is it is finally cleaving your arginine into ornithine and urea. Okay, ornithine and urea. So the final step of urea is it is basically composed of carbon dioxide. So can you see, it's basically composed of carbon dioxide and your ammonia, okay, and the amino group of aspartate okay. So this structure is actually water soluble. So this structure is water soluble and is going to be transported very easily through the kidneys, and therefore for the excretion through the bloodstream. So what is the important role of amino acid metabolism in the whole urea cycle? Your amino acid is going to catabolize your ammonia. Which is toxic to the body. Ammonia is then converted into urea by the urea cycle and excreted. So I think I already told you about how the ammonia is transported, right? That is how it is going up above. I think last class I talked about your ALT and everything. So that cycle is a little bit down. So this is the transport of ammonia by your alanine. Last class I discussed about this. Okay. So your ALT levels are measured to determine your liver function, right? So why is the ALT levels measured? For your liver function. So another transaminase is your aspartate aminotransferase. Okay. Aspartate aminotransferase is actually measured in conjunction with your ALT. So whenever you're having a hepatocyte injury, whenever you're having a hepatocyte injury, what will happen? These enzymes are going to be released. Okay, so you will have high levels of these enzymes. So when you're having high levels of these enzymes, that means we're having a liver damage. So the ratio between your AST and ALT is very important for the cause of the liver disease. Okay, so most of the liver diseases will have a greater increase in your ALT than your AST. That is, your AST ALT ratio will be greater than 2:1. Okay. So if it is going to be greater than 2:1, then it is going to be a alcoholic liver disease. Okay. But otherwise, in a liver disease, your ALT is going to be higher than your AST. Okay. Your ALT is going to be higher than your AST. So how do you think diet is going to affect your urea cycle? So here, if you see certain enzymes, okay, high protein diets, they will increase the concentration of certain enzymes, okay, specifically your ornithine trans carboxylase, okay, and your arginosuccinate synthetase, okay, and your arginosuccinate lyase, okay. So these concentration has been increased when you take a lot of protein. Okay, so that is low protein diet is obviously going to have the opposite. So what happens in a starvation condition? Starvation will upregulate your urea cycle. Starvation will upregulate your urea cycle. You will have increased urea excretion. You will have increased urea excretion. This because fasting will cause the body to break down protein. No. When you're fasting, where will the energy come from? We have to break down the protein only. So that's going to create a lot of ammonia. So that's why your urea cycle is being upgraded and you're going to have a lot of urea excretion. Okay. So that's about your diet and urea cycle relation. So what are the disorders that you can have in a urea cycle?
Ornithine transcarbamylase.
Yeah.
OTC. Hyperammonemia.
Very good. So all the urea enzymes that I have, yeah. So all the urea enzymes that I mentioned, right? So you will have a disorder. Okay, all urea enzymes will present with hyperammonemia. Okay, so I think I will make a tabular column for that so that you guys can understand it. So first is we are having a hyperammonemia. Okay, so this hyperammonemia can either be acidosis or it can actually be of normal pH or alkalosis, right? So now, if it is going to be acidosis, then you have to think about organic organic academia, or your pyruvate metabolism, Okay, so if it is going to be normal or alkalosis, then you have to see the sugar, okay, whether the sugar is normal, that is normoglycemia, or it is hypoglycemia and hypoketosis. So you have to see whether these things are present. Okay. So then if it is going to be normoglycemia, then you have to look into your amino acids. Okay. Amino acids. So in that you will see, okay, whether the citrulline, okay, whether the citrulline is increased, okay, and the ASA is absent, okay. Otherwise, whether the citrulline is increased and your ASA is present, okay. Otherwise, you see if the citrulline is low or it is absent, okay. So if it is going to be a low citrulline or a absent citrulline, okay, if it is going to be, so till now just write it down so that I can have space. Just let me know when I can rub it off.
Done, ma'am.
Okay, others?
Ma'am, could you please explain this again? The call disconnected for me, unfortunately.
Oh, okay. So when you have a hyperammonemia, so I'm talking about how do you go about why it is happening. So either you can have a hypoammonemia and a acidosis, or a hyperammonemia and a normal pH or a alkalosis. Okay, if it is going to be acidosis, then it is your organic academia or your pyruvate metabolism. Otherwise, if it's going to be your normal pH or your alkaline alkalosis, then you have to check your sugars. So if it is going to be a normal sugar, then you have to check your citrulline and your ASA. So either the citrulline is increased or decreased, or either the ASA is absent or present in your combination of increased citrulline. And then you have a decreased citrulline or a absent citrulline. And then the last option is decreased sugar, that is hypoglycemia, and then you can have hypoketosis. So I'm going to continue this only, but I need a little bit more space. So can I erase?
Yes, ma'am.
Okay, fine. So now acidosis, I already told you that it is organic academia, right? So now coming about first condition, that is your your hypoglycemia and hypoketosis. So hypoglycemia and hypoketosis, you have to think about fatty acid oxidation. Fatty acid oxidation, okay? And then next is your increased citrulline and ASA present. Okay, increased citrulline and ASA present means you have to think about your argininosuccinic aciduria. Argininosuccinic aciduria. If it is going to be a low or a absent citrulline, if it is going to be a low or a absent citrulline, then you have to look at the orotic acid or your orotidine. You have to look at your orotic acid or your orotidine. If your orotidine is normal or low, or if your orotidine or your orotic acid is high, if it is going to be high, then it is a OTC deficiency. And if it is going to be a normal or low, it is going to be a CPS deficiency. The enzymes that I'm talking about, no, I told you all enzymes will cause hyperammonemia. So how will you find out which is a specific enzyme? That's why this is important. And next is your increased citrulline, but you do not have ASA. ASA is absent. So that's when you call it as citrullinemia. Okay, citrullinemia. So depending upon the plasma metabolites, you go for each and everything. Okay, so or you can make a tabular column out of this also. I will give you a tabular column. I hope that tabular column is there in the first stage. And first aid 2022 version has been released. Okay, so that is available in the Telegram, but I wouldn't be able to send in the group. It's approximately 243 MB. You can I'll just send the link, download it for yourself. The images alone have been changed, but I haven't checked the entire textbook. Okay, I will erase this. Okay, the tabular column is not given. So you need this. Okay, you have to know N-acetyl, or I'll just send you the tabular column as a drawn thing. Okay, because it's a very big tabular column. It talks about you will have a short thing about your enzyme that is deficient and what is the inheritance of it and how will you talk about your metabolite levels. Okay, so I'll make the tabular column and I will send it to you. So now coming to the other important things about your urea cycle. Okay, so I already talked about how your hyperammonemia is going to affect the brain, but any idea how your urea cycle and the pyrimidine synthesis, how it is similar?
Carbamylase CPS enzyme.
Yeah. So if
There
you
is
look at,
CPS2 in the pyrimidine cycle, pyrimidine synthesis, and we have CPS1 in urea cycle.
yes. So if you look at your urea cycle and if you look at your pyrimidine synthesis, okay. So if there is a CPS1, there will always be a CPS2. So what is the difference between them? Your CPS1, that is the one that is synthesizing your carboxyl phosphate. Okay, your carboxyl phosphate synthesis by CPS1 is in the mitochondria in the urea cycle. Okay, so the carboxyl phosphate is an intermediate. Okay, is an intermediate not just for urea cycle but also in the pyrimidine synthesis. Okay, because it is happening in the cytosol, CPS2 is the isoform of CPS1 that can produce carboxyl phosphate only, but it can produce carboxyl phosphate outside the mitochondria. Okay, that's the similarity. And then what other things? I already taught you about the orotic acid deficiency, right? I remember one class I mentioned about it. So whenever you
Yes,
have
ma'am.
a CPS, yeah, whenever you have a CPS1 deficiency and when there is a low or absent citrulline and when there is a normal levels of orotic acid, you have to think about that. Okay. So OTC deficiency also, I will give you the tabular column that will give you about the inheritance. So other than that, in urea cycle, nothing else. So I will ask you guys questions. 48-year-old homeless man is presenting with confusion and is also presenting with slurred speech. Okay, so now we did a physical examination that is showing spider angiomas. It is showing spider angiomas where on the chest, and the patient is also having flapping tremors. The patient is also having flapping tremors on the wrist. Okay, when he is extending it. Wrist extension. Okay, so now we are doing a lab test. It is showing elevated AST and it is also showing elevated ALT. Okay, so where do you think the first step of this patient's metabolic defect, the first step of the patient's metabolic defect is occurring? Maybe I'll repeat. A 48-year-old man having confusion and slurred speech and spider angiomas and flapping tremors, increased ASE and ALT. Okay, so where do you think is the first step of the metabolic defect is occurring? So when I started the cycle, I said something, right? Okay, so the first two steps of the urea cycle occur in the mitochondria. Okay, so it is hepatocyte mitochondria. The final three steps are occurring in the cytoplasm. Okay, so this is just a clinical presentation, but the question is not about the clinical presentation. The question is just about the biochemistry. Okay, again, I'm repeating, whenever I'm giving you a case, listen to my last question alone. Okay, even when you're solving a USMLE case, listen to just the last question and then you have to find the clues regarding that question because you will have a lot of unnecessary things. Okay. Next question. Which of the following metabolite levels. Oh, I think I will not ask this question because I didn't give you the tabular column. I will change the question. So patient is actually or say a research person, okay? A researcher is comparing the effects. A researcher is comparing the effects of a high protein diet on the rats who are having a OTC deficiency, and the rats who do not have a OTC deficiency. Okay, so after the high protein exposure, after the high protein diet or the high protein exposure that they're getting, so the rats who are having a OTC deficiency, they're having a increase in the brain levels of which substance? They're having an increase in the brain level of which substance?